All right. So I think that Simon's lecture today will actually perhaps give you some ideas then for some things to try with your program, because he's got a really interesting application today for the skills that you've been learning so far. That's very firmly within the realm of what you would actually do with data you get from the lab. So with that, welcome to this last week of our R for Biologists workshop. What we're going to do is Simon's going to give his lecture, and then at the end, we're going to talk a little bit about next steps. But for now, Simon, you want to go ahead and take it away?
Yeah, I'm going to try sharing my screen. Okay, cool. So, welcome to this last week. I'm going to get started right away so that we have time to talk at the end. So today we'll recap last week's tidyverse basics that we went over, and I'll just briefly remind you of the basic concepts from last week, and then we'll move on to the last bit of the Titanic data set, where we're going to use a package called ggpubr that works together with ggplot to incorporate statistics onto your plots and make them publication ready. And in the end, I'll show you how to apply the tidyverse and ggpubr to biological data from actually my thesis project. And it's just a simple plate reader assay that I have luminescence data from a plate reader, and we're gonna analyze that data with R. So just to remind you, we're working with the Titanic data set in the beginning that contains various things concerning all the passengers on the Titanic. And the tidyverse is based around the pipe operator. So this, let me get my pointer here. This guy right here that works to pipe a piece of data as the first argument to a variety of functions, so that you don't have to do this here. You don't have to use intermediate data frames. You can just have a pipeline to analyze your data from start to finish without intermediates. So some commonly used verbs. Remember that verbs are the name that we give functions when they're in a tidyverse pipeline. There's filter, where we filter rows. There's select, where we filter columns. There's group by, where we group the rows by the value of a certain variable. We can summarize, which with the British or American spelling, where we summarize a group, a piece of data in some way. Here, the n function just counts numbers of rows. So if we group here by sex and survived, this will count the numbers of each sex survived combination. Mutate creates a new column in the data frame based on some manipulation of existing columns. So in this case here, we're making a percentage calculation, but this can be anything you want. And then filter again. So you'll see these guys pop up again and again today, and then we'll have some extra ones just to show you that just because you know the basic ones doesn't mean you'll be able to do anything, but it's always just a Google away to find a new verb to do what you need to do. Then ggplot is based on the grammar of graphics that builds plots by layering different elements of a plot, such as data, aesthetics, geometries, and so forth. That's represented here. And so some commonly used layers in a plot. So this is from last week's lecture as well. We have the ggplot basic function where we specify the data and the aesthetics, so the axes that we want. We want an x-axis, we want a y-axis. Here in this case, we have a fill aesthetic as well. So we're going to color our bars in this case by another variable. And then we add new layers with the plus. So no longer the pipe operator, because this is all the layers of one plot. So we're going to add things with a plus. So the geometry here is going to be a bar plot. We can do labels for the y-axis here. We can do a title. We can set up the visual theme, x-axis label, and so on and so forth. We can scale our axes differently. So these are some commonly used layers in a plot that you can kind of use as a very, very basic reference for when you make your own plots. All right. So if you'll remember, this was the last slide basically from last week's lecture where we did where we read the Titanic data in, and then we filtered and selected various things to get an analysis of the percentage of people who survived within sex. So what percentage of women survived, what percentage of men survived. And so we filtered by age to only look at not exactly adults, but not children anyway. We selected the survived and sex columns, grouped by them, counted the number of people in each category, made it into a percentage, looked at only the people who survived, and then plotted it with the data layers that I showed you in the previous slide. And so that's what that looked like, right? So you'll remember this from last lecture. So that's just a bit of a recap of how a really quick run through of like an end-to-end pipeline, just so you put your mind back into the right frame. So we're going to now move on and add one last piece to this construction that starts with data manipulation, data plotting, and then the last piece is statistics on that plot and the plotted data. So what we're going to want to know here is, does FAIR predict survival? So is it true that the more you paid to get on the Titanic, the better your chances of surviving were, or is it not? And so we're going to use the same Titanic data set. We're going to get it from GitHub, and we're going to read it into this new variable that we're going to call Titanic, so that we don't have to have this long URL at the beginning every time. And so let's just try and plot this right away. Let's just give it a shot and see what happens. If we just say we want survived on the x-axis and FAIR on the y-axis, and just see what there is to see, right? And so right away, you notice a problem. Survived here is coded as 0 for didn't survive and 1 for survive. But here it's 0.25, 0.5, 0.75, and then 1. So the problem here seems to be that the survived variable that we're plotting on the x-axis is considered as a continuous variable that can take any value between 0 and 1, which is, of course, not true. And you'll see that here, when you look at just the survived column, we see that it's a double, which is a decimal number, so not an integer number. So this is continuous. So we don't want that. We want this to be discrete, because it's either 0 or 1. You can't half survive. So what do? We turn survived into a factor. Now, if you'll remember your R basics, and this is why we teach you the R basics in the beginning and the data structures, because it's important to have an understanding of them and be able to apply it later when problems like these crop up. So factor is like a vector, except that it is ordinal. So it has discrete values that you can order if you so choose. So when we turn survived, a vector that only contains zeros and ones, into a factor, it'll now mean that R behind the scenes will now know that the elements of survived can only take the values 1 or 0. And that is exactly what we see when we plot this factor now. It's now a 0 here and a 1 here. There are no intermediate values. There's nothing below 0, but nothing above 1, and no 0.5s. So we don't want this to say 0 and 1. We want it to say died and survived. So when we create this factor, we can add an extra argument that's called labels. It'll do this automatically in order, so 0 first and then 1. And we're going to call them died and survived. This is just for our understanding. This has no influence on the program itself. And so now when we do this, it'll say died here and survived here. We have this somewhat ugly x-axis label. We're going to deal with that later. But now our x-axis is correctly coded. So next, last time we used a bar plot. This time we're going to use a box plot. And that is what that looks like. Now, this isn't very informative because the vast majority of the data is squished here in the area of the y-axis that's between 0 and 100, because apparently most people paid that amount for their fare. But there are a few really high ones, like up here above 500, and a few like intermediate high ones. So what do we do when the values in our y-axis span a large range of values, and it's hard to get distinctive information from it? We turn it into a logarithmic scale. So here we can use the scale y log 10 function to turn the fair y-axis into a logarithm. And now our box plot suddenly is visually a much more powerful. We can tell that there appears to be the possibility for a difference here. The statistics will tell us if it's significant, but now at least we can see that the median here, they appear to not be the same. All right, so once we do this all live in R again, like we did last time, you'll see that this will throw you a warning that it filtered out some of the data, and that is because some of the rows don't contain fair data. It's a 0, and so when you do a logarithmic y-axis, that'll throw you a problem because the logarithm of 0 is not defined, and so it'll filter that out. And to avoid this warning, we're just going to, before we do all of our plotting, we're going to filter out anyone whose fair information is 0, just to use cleaner data. And again, the plot won't change, but we won't get the warning message. So now we want to do stats, right? Because until now, this is the same thing we did last time, except we used a box plot instead of a bar plot. So now we're going to add statistics to the mix. So we're going to load the ggpubr package, and we're going to add a t-test. You do this with the stat compare means function, and then this function takes an argument called method, where you can specify what kind of test you want to run to compare means. In our case, this will be the t-test. And if we do that, it'll display at the top here, it'll say it'll do the t-test, and the p-value is smaller than 2 times 10 to the minus 16. So very significant by almost any standard that you could reasonably have. This will be very significant, so we can be very confident that there is a difference in means here. However, those of you who are familiar with doing papers will know that this is not a very visually appealing way of representing the results of your statistics. It is good because it gives you the actual p-value, but what we want is the little asterisks. So this is what you can do with a label argument here, and the p.signif value of this label, of this argument, will make it so that we have stars. Now we have 4 stars here. But what we really want is a nice little bracket that tells us that we're comparing this with the stars centered in the middle. So that is what we do with the comparisons argument. Here we can specify a concrete list of comparisons. Now, comparisons takes a list. This argument takes a list as a value. So we need to make a list of vectors of variables that we want to compare. So C makes a vector, if you'll remember from your basic R. So in this case, we only have the one comparison, right? So we have C, died, survived. This is our one comparison, and we make that into a list. So that list will have one element, and the single element that this list contains is a vector of length 2 that contains the variable names died and survived, or the variable values died and survived. If we were comparing a group of 16 means with an ANOVA, we could add more vectors to this list to specify what post hoc comparisons we want to make. So when we do this, that's what that looks like. And this is what I'm sure we're all familiar with from reading papers and from what Prism spits out. So this is how you add in statistics to your analysis. In the end, we're going to add a couple of more layers just to improve the overall appearance. We're going to make the X label null, so we're going to take that away because it already says died and survived. We're going to call the y-axis ticket price. We're going to make a black and white theme, and then we're going to do ggsave at the end. We're going to call this file myfigure.png. We're going to give it a height and a width of 5 inches, and that will be saved. That will save that figure as an image that looks like this. So before we move on to biomedical data, let's do this in our studio. So we're going to start by loading the tidyverse.
Simon, just really quick, which file is this one?
Sorry, this is the tidyverse _week6.r.
Okay, so you should have all at this point gotten the GitHub, right? The zip file from the GitHub, extracted that, and that should, when you open that up, if you open up the tidyverse week 6, that'll be what you're looking at here. Okay.
All right.
Put it in the chat if you're not able to find that, and I'll help you.
Okay. I'll give you like 30 seconds just to open that in case you haven't.
Hey, Simon.
Yep.
To the point where we're waiting on that. So when you just did the comparisons right now, you under comparisons you listed died and survived. Let's say, for example, if we had one more bar graph, and I wanted to indicate similar comparisons, how would we change the code line?
Right. Okay. So I'll do this in the console, right? So the thing that we did here lists the value of the comparisons argument was this. So I'll do this right now, and I'll actually call it something. Now we have comparisons. It's a list that has one character vector, right? So it's actually easier to do like this. So now, if we had more than that, we would simply add more vectors to this list. That's why it's a list and not just a vector, right? So we have C, group 1, group 2, and then we also have C, group 1, group 3.
Oh, okay.
And now, you see, it has 2 elements.
Sure.
The list has 2 elements, each of which is a vector of length 2 that specifies a comparison. So this would be a classic done its post hoc test setup, right? You have G1 is the control group, and you have 2 treatments, G2 and G3.
Gotcha. Okay. So you just need to list the comparisons and introduce them as string characters.
Yep. Correct.
Okay.
And the strings here, they have to be what those values, what those groups are called in your graph.
Correct. Okay. Thank you.
All right. I hope that gave everyone time to load up the right script. So what we did here is we loaded the tidyverse, and now we're going to read the CSV file from this URL into a new variable called Titanic. I'm actually going to clean this up so that we can see variables as they appear. So now we have the Titanic data frame that we should be familiar with by now. All right. So like I said, we're going to start by just plotting it. And then we see here this problem that it's, in fact, a continuous variable as evidenced by the double type here. So what we're going to do is we're going to plot it as a factor. And now it's it's discrete. We're going to add the x-axis labels, died and survived. And we're going to add the box plot geometry layer. Now you'll remember that this was not visually appealing, and so we're going to change it to a logarithmic y-axis. And now this is what I was talking about. Don't mind the German here. It says transformation introduced infinite values in continuous y-axis. What does that mean, right? The transformation was that we scaled our y-axis into a logarithm. So our transformation was to take the base 10 logarithm of each y value. Some of the y values were 0, apparently, because the log base 10 of 0 is, well, it tends to infinite, but it's undefined. So it removed 15 rows containing infinite values. And while it didn't do anything bad to our plot, it just omitted those data. We don't like that. We don't like this dirty data. So like I said in the PowerPoint, we're going to introduce this line here at the top of our analysis to filter out anyone whose fare is listed as 0. And so now we get the same plot, but we don't get this warning anymore. So next, we're going to load the ggpub library. If you might have installed it last time. If you didn't, it would be this command right here to install the package. So we're going to load ggpubr, and we're going to add the t-test. As before, this lists the t-test t-value up here, We're going to turn it into an asterisk. And then additionally, we're going to list the specific comparisons we're making so that it can be visually represented by the bracket at the top of the graph. And then we'll improve the aesthetic of the graph and save it as a new file. So if you do all this and go to your folder, whatever folder this is happening in right now, that will be displayed at the top here, you should now have a file called my_figure.png that is that figure. So if I actually go to my one right here, I have a myfigure.png that looks like that. All right. So that is everything I'm going to show you about how to use the tidyverse to analyze data. The next step will be to apply this to a biomedical data set from a plate reader. Before I go there, does anyone have any questions about all the theory we've just gone over? Great. If not, then I'll switch back to the PowerPoint. But what I'll be going through in the PowerPoint is what's going to be contained in this luminescence.r script. So you can follow along in the PowerPoint or in the script. Like I did just now, I will, once I'm done with the PowerPoint, switch back to the script and we'll do it live so you can see how it all works out in RStudio. All right. So just to give you a bit of an idea of what exactly it is that we're looking at here, this is the elevator pitch for my thesis, for my PhD thesis. So I work on a group of diseases that are called tauopathy, where the main characteristic is that a protein called tau aggregates. And the purpose of my thesis is to develop a system where we can use luminescence as a readout to quantify the extent to which tau aggregates in a fruit fly model, as it happens for me, but this technology is applicable to neuron models or cell-based models also. So the way it works is a split luciferase assay. So what we do is we split the luciferase protein that makes fireflies glow in the dark, so it emits light in the presence of its substrate. We split it in half, the C and N terminal half, and we fuse each half to the tau protein. Now, when tau aggregates, like shown here, the C and N terminal halves of luciferase will come into really close proximity and partially reconstitute activity of the luciferase enzyme. When you then feed these flies the luciferase substrate, the partially reconstituted enzyme will produce light, and that light can be detected in a plate reader. I also have a control construct where instead of fusing the luciferase halves to tau, I fuse them to GFP, and GFP should not aggregate, so there should be at least, if none, then at least less luminescence from that. So what I have is a plate reader experiment right here, where this is what the Excel file looks like that the plate reader gives me. Row A was the tau aggregation flies, they're called tau ag, and then rows B and C are two different clones of the GFP control construct. And the goal of the experiment is to find out which of the eGFP controls works better as a control, so which has lower luminescence. I'm trying to find the best control to move forward with in my project. So I have three groups, and I'm going to be comparing their means. That should start ringing bells with everyone as far as what kind of tests we're going to be doing. Questions about this? Cool. All right. So we're going to start by reading in the data. We're going to read it in from a CSV. In the script, I've also given you instructions for how to read it in from the Excel file, but generally CSVs are a bit more straightforward. So we're going to call our data frame luminescence, and this is the CSV file that should also be in the GitHub that you guys should have downloaded. And as long as you're downloading it all to the same folder, this line will just read that CSV file into a data frame. And if we display the data frame, this is what it looks like. So what we have is, I'm showing you the first 5, but we have 18 rows. Each has this ID string, a sequence ID, neither of which we care about. But what we do care about is the well position, right? So this is well A1, this is A2, A3. This is a standard 96 well plate. So you can picture what that looks like. Then we have 2 logical scan position and pack that we don't care about. And we have RLU, which is our relative luminescence units. So this is the actual measurement that the plate reader made for each well. And then we have two more QC metrics here that we don't really care about. So really what we care about is this well position and this RLU. So you'll note that there is no information in this data frame as to what each well is, right? The identity of the samples in each well, because R doesn't know. R just knows this was well A1. This is all R knows. So what we have to do in order to do this analysis, we have to make our own data, our own secondary data frame. I'm calling it plate_layout here, to tell R what the layout is of our plate. And I'll show you how to merge this plate layout data that we're coding here right now with the luminescence data in a minute. But we have to, that's just the one thing we have to do manually. We have to manually make a layout that we can then use that information that's contained in it. So the data.frame function makes a data frame. And what we do is we specify rows. So what we do is we specify columns in the data frame. So we specify them by giving them a name and the values. So my first column, somewhat ironically, is called row, but it makes sense because it's just what row am I talking about here, right? So this is the output of the command up here. So this is the data frame that I'm creating right here. So it has 3 rows, and it has 2 columns. The first column is called row. It contains the values A, B, and C. And then the second column is called genotype, and it says that row A, these 6 samples here, are the tau ag genotype, so the tau aggregation flies. And then rows B and C, these samples here, they are the 2 clones of the EGFP ag, the 2 controls that I'm trying to see which is better. So I have one variable called row that contains the values A, B, and C as strings, and that will get populated by row like this. And then my second column, my second variable is called genotype, and it takes these 3 strings, and as you can see here in the resulting data frame, it gets populated from the top down like this. Does anybody have a question about this? This is like the newest thing. This is like the one new thing that wasn't part of the previous analyses, because we have to manually supply some information. Great. So you will remember that the well position, I'll do it like this, is A:1, A:2, A:3, right? So what we want is we want to split this into just a row and a column. We don't want this colon in between, and we want them to have the row and the column to be separately accessible. Now, as I mentioned, the functions and verbs I've shown you so far, they will not cover every use case. For example, I've never shown you how to split a character string into its parts, because you don't do that a lot, but sometimes, such as in this case, you do have to do it. So what we do is we go and Google how to do that. And I guarantee you, if you Google R, how to split strings into characters, this substring function will show up really quickly. How does it work? It takes a string as an argument. We're supplying the well position string. And then you're telling it what to cut out by start and stop position. Now I only want the one letter, so I start at the first letter, and I stop at the first letter. So that will take the letter A here, 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 and then the letter B from here to here, and the letter C from here to here. I do the same where I mutate for a new variable called column, where instead of 1, 1, I do 3, 3, because I start at the 3rd character in the string, and I stop there too. So 1, 2, 3. It'll be the number here. So if we look at the 2 new variables that I'm creating with this command, I'm creating a variable called row. The first 6 samples are A, the second 6 samples are B, and the third 6 samples are C. And then I have the corresponding column numbers here. Next, this is going to be more familiar. I'm selecting only the relevant data, because you saw there was a bunch of data in there that's QC that I don't really care about. So I'm taking the RLU column. This is how you rename it when you select. I take the RLU column and I name it luminescence, because that's what it is. And I take the row and the column. So this is my new data frame. Now I just have row, column, and luminescence. This is all that I need for my analysis. This is all that I'm working with right now. Next is where we introduce the manual data frame that we made a minute ago. We want to fuse together two data frames. We want to bring the genotype information into this data frame. So this is what you do with a join. There's left join, right join, outer join, and inner join. They are all slightly different, and you can Google the differences. They are not very difficult to understand. In this case, any of them would work. They only matter when you don't have a good match, when you have a one-to-many match or a many-to-one match. So what we do is we join the luminescence data frame to the plate layout data frame. And the one argument we have to supply is the by argument. So what are we matching them by, right? And what we are matching them by is the row, because our layout data frame contains rows matched to genotypes. And our luminescence data frame contains a bunch of samples that each have a row value. So when we do that, the tau ag genotype that goes with row A gets added to each row in the luminescence data frame where the row value is A. Same for the row value B. They all get the EGFP ag 1. Row value C, they each get EGFP ag underscore 2. Does that make sense to everyone, or anyone really? Now is the time to ask questions. Okay.
Can you go over that one more time, please?
Yes. So you have your luminescence data frame that's just this on the left, right? That was just this.
Yeah.
And we have a layout data frame that has
Genotype.
3 rows and 2 columns. The 3 rows are A, B, and C, and the columns are row and then genotype, right? So so the layout data frame matches row letters to genotypes. Does that make sense so far?
Yeah.
Okay. So now what we want to do is we want to combine the two, right? So we want to join the two data frames so that anything with a row letter A in our luminescence data frame over here will get the corresponding genotype, tau ag. Everything with the row letter B will get its corresponding genotype, EGFP ag 1. And row letter C, the same, EGFP ag 2. So this is what this left join does. We merge together the two data frames, and this by equals row tells it that we want to match the data frames up by the value of this right here, because both the luminescence data frame and the layout data frame have a column called row that contain the elements A, B, or C. So you can now match them according to that. Does that make sense?
Yeah.
Okay, and that's all it does. So now every time you have an A in the row column here, it'll join
Tau AG.
the layout that was in the row that was in the A row in the other data frame. And every time the plate layout data frame's row says B, it'll take EGFP ag 1. And every time the plate layout's row column says C, it'll take EGFP ag 2.
Okay, thank you.
All right. Next, we group by genotype, and we calculate the mean within groups. And we add it as a new column. We could do this with summarize, but then we would lose all the rest of the data. We would lose the individual values, and all we would be left with is a data frame that just says the group and the mean. But we don't want to do that. What we want is we want a new column that contains the mean, and we want to retain all the other data. So we're going to use mutate instead of summarize. We're going to make a new column called luminescence mean, and it's going to be the mean of the luminescence column. Because we grouped it here by genotype first, the mean will be calculated within group and not across the entire data frame. So as you can see, everything with the genotype tau ag has a mean of 2000. Everything with a genotype EGFPAG1 has this 1386 and then 801. The next thing we do is calculate the standard deviation. In the same way, we take luminescence underscore stdev, and the SD function is what is the R function for calculating standard deviation. And it gives us this new column where within each group, we've now calculated the standard deviation. And lastly, because we're going to be plotting means, we actually want the standard error of the mean instead of the standard deviation. So we take the standard deviation that we've just calculated, and we divide it by the square root of the number of rows within each group. So this is where the stats comes in, right? You have to know your stats. The standard error of the mean is standard deviation divided by the square root of your n. Now your n, you access it by counting the number of elements in each group. And that is what that looks like. 230 here, 104 here, 107 here. We good? Everyone with me? Cool.
Yeah.
All right. So we're done. We're done wrangling our data. We have now grouped it. We have the individual data points. We have the mean, the SD, and the SEM. All that's left to do is plot it and do the stats. So ggplot, X is a factor of the genotype because genotypes aren't intermediate. You're one of these three genotypes, and that's it. So we make a factor out of the genotype column, and we specify the levels. We call them tau-ag, EGFPAG1, EGFPAG2. And on the Y, we're going to plot the luminescence. Now, note that I'm plotting the luminescence here and not the luminescence mean. Visually, that won't matter just yet, but behind the scenes, the individual data points will be retained. So we can do stats on them, because if you only provide the mean, it can't do any stats on the mean alone. So we have a graph, empty so far, with luminescence on the y-axis and our three genotypes on the x. Next, we add a bar graph. We could do this with a box plot, but I chose a bar graph because that's more commonly used for biology graphs like this. So if you remember from the Titanic, the bar graph wants a stat argument. You can use identity, and then it will do nothing, but that's what you would want to use if you plotted the mean. Now, because we provided the individual data points, not the mean, you have 6 data points behind the scenes for each group, right? Because we just gave it this. So what we want to do is visually, the bar visual needs to plot the mean of each group, not each data point, but the mean. So for the stat argument, we say summary, which says we want to provide some function to summarize the data. And we specify which function with the fun argument. And we say it is mean. And so it'll look like this. It'll plot the means of each group. Now let me go over that again. If I could give you visually the same plot if I had said luminescence underscore mean here, because I've calculated the mean, right? I could give you visually the same plot by saying luminescence underscore mean here, and then instead of all this, I would just say identity here for the stat argument. And it would look just like this. But we couldn't do stats with it because behind the scenes, all of the individual data points would be gone and R couldn't do math with them anymore. So instead, I gave it not the mean, but all 18 data points and told them to summarize each group by the mean so that we get a graph that looks like this. The next thing we want to do is error bars. So this is where the mean and the SEM come in. We add the geom error bar. This is a new thing, but it works just like geom bar. We can add another layer of visual information that is an error bar. So it takes 2 arguments, y-min and y-max in the aesthetics here. So what is the minimum, the minimum y value of our error bar? Well, it's the mean minus the error. And the maximum y value is the mean plus the error. So when we do this, it looks like this. We have an error bar that's centered at the mean, and it extends The error, it extends below the mean by the error, and it extends above the mean by the error. So this is all the graphical information about our data that we want. The next thing to do is to improve the appearance a little bit before we move on to the stats. So if you recall, geom error bar is the last thing we did. So I'm just going to change the X and Y axis labels. I'm going to give the graph a title. I'm going to give it the classic theme. And it'll look like this. It's a little bit easier to take in visually. So now we want stats. So we have 3 means instead of 2. And we want to see if there is a significant difference in means. Now, what statistical test do we use for this? Anybody?
Nova.
Correct. Oh, chat. Probably someone saying ANOVA in chat, I'm assuming. Okay. So we add the stat compare means, just like before, but instead of t-test for the method, we now say ANOVA. This label Y just says where on the graph I want the visual text to be situated. So now it has done an ANOVA for me, and the p-value is 0.00032, which is significant. So we have a significant effect of genotype on the mean of luminescence, which is great. Next, what we do is we add the post hoc. Now, here I've done something that all of you will want to kill me for, and you should. I made post hoc t-tests, which you should not do. The reason I did this is because one of the downsides of ggpubr is that it does not easily incorporate ANOVA post hoc test. You can do it, but this would require me to give you, to introduce you how to do ANOVAs outside of ggplot or ggpubr and how to feed that information back in. You can totally do it. If you'll just Google it, you will find step-by-step instructions and actually code that you can just copy and paste. But for the sake of simplicity and just showing you that you can do an ANOVA without diving too deep down the rabbit hole, I did post hoc t-tests knowing full well that this is not what you should do mathematically. But, and this will answer Mustafa's question also for how you add more comparison. I have another stat compare means, this one using t-tests, and my comparisons here is a list of 2 vectors now. One comparing tau-ag to EGFP-ag1, and one comparing tau-ag to EGFP2. And I gave it the label p significance so that it displays the asterisks instead of the actual test statistic. And in the end, I saved the whole thing as raw_luminescence.png. And that is what it looks like. So both are significantly less than the tau-ag, which is good because GFP should not aggregate. Neither is 0, which means that there is some background signal coming from my system. But the second one here, the GFP-ag2, seems to work better in that it has less luminescence. And that is what I chose, in fact, to use forward with in my actual dissertation project. I can tell you I did this with a Dunnett post hoc test, and the significances are the same. Like, it just so happens that the t-test gave me the same result here. But we all know, don't do post hoc t-tests. Are there any questions at this point?
I have a question about that mutate function.
The mutate function?
Yes, so when you applied that function, how come it only calculated mean standard deviation and the error within that particular group and not ABC altogether?
Yes, this is because I did the group by up here, group by genotype. This behind the scenes groups the data frame by whatever variable I provide, and in this case as a genotype. And so whenever I use summarizing functions like mean or SD or n, it will only apply them to the last level of group that you supplied.
Okay, thank you.
I want to just really briefly point out that this is different from using the summarize function, because the summarize function will actually remove your grouping. Mutate won't remove your grouping when you run it.
Yep, and it also won't remove the individual data that you used, right? So I still have access to the individual data points. Summarize will just spit out a data frame that's ungrouped that'll say genotype, mean, genotype, mean, genotype, mean, without retaining access to the individual values that make up the mean. All right. Now, I want to actually not plot it like this. This is all fine and dandy, but really luminescence values from a plate reader are arbitrary units, so it doesn't matter if this says 2,000 or 20,000. All that matters is the relative difference. So what do? We normalize. We normalize to control, which in this case is tau A. How do we do this? So I'm showing you the whole thing that I just did, except I changed a couple of things. Before grouping, I introduced one mutate call that makes a new column called luminescence norm, where every single value of luminescence gets divided by the mean of all the values of my luminescence data frame, where genotype equals tau A. So this is a bit of an advanced technique right here, to use this this test of whether something is equal to something else, as a way to subset your data frame. So my luminescence data frame here, the brackets will allow me to select certain rows of that data frame. And I'm selecting all the ones where genotype is equal to tau A, which will be 6 rows. And I'm taking the mean of that. And every value of luminescence, no matter what the genotype, will get divided by that mean. Then I do the same for the entire rest, except every time I did something with luminescence, like calculate the mean, standard deviation, and SEM, I now do it on the luminescence underscore norm. And the plotting gets adjusted accordingly, where I plot the normalized luminescence value, and the error bars are at the mean and error of the normalized luminescence instead of the raw luminescence. What this one extra line and these couple of adjustments here to make it all fit does is it gives me a graph that looks suspiciously similar, except it is now centered around 1. So now I have normalized. All right. I'm now going to switch over to RStudio, and we can do this live. And if you have questions, you can ask them while I do them live, so that I can show you directly how it works. Does anyone have any burning questions right now before we get going?
It might be good to mention, because I don't think there's an RStudio project in there, might be good to mention briefly how you get to the working directory. I don't know if we've actually talked about that before.
No, we haven't dealt with sort of projects and working directory setups at all. But I mean, your working directory is by default. Sorry.
Oh no, I was just gonna reiterate on Henry's point. I couldn't read the luminescence file, so I had to change my working directory to make sure that the R was reading it from the correct directory, so it could read luminescence files.
Okay.
Mm-hmm.
Yeah.
So perhaps the easiest way to do this, if you're not familiar with sort of how file paths work in your system, is to just make an R project in the top right. And you can just place that R project in the directory that the data is in.
Yep.
Actually, yeah, maybe we could, because that just takes a second, maybe we could show that really quickly. Right, so see where he's doing new project. And yeah, that's yeah.
I'll make a new directory.
You would go to existing directory. Yeah, and then you can just search. If you go to browse.
I mean, it's already at the right one.
Yeah, it's just, this is for most people it won't be. So if you just browse, look for where you downloaded the GitHub zip file, and just click on that module 6 folder, and that'll be, you can just hit open. Yeah.
So, yeah, I would have downloaded it and extracted it here. So now I have the module 6. Double click and then do it here.
Right. Yeah. And if you create it there, and if you open that up there, then all the paths that Simon has in the code will work exactly right.
Yeah. So now my project up here says module number 6. And I have all this data all this stuff in my project here. So I can open luminescence.r from here.
I kind of discourage people from ever using get working directory or set working directory, because when you give someone else your code, it won't work the same for them. And so our projects are a nice way of giving someone else your code, but not having to worry about them having to set their file paths and that sort of thing.
That's true. I'm just going to reload the packages real quick. All right. All right, let's do this. So like I said, first we're going to read in this CSV. And when we look at it, we see that it's 18 rows. And what we care about here is the well position and the RLU. This commented part here is how you would get the same data out of an Excel file. Now, it depends a little bit on your individual plate reader, the format that they spit out the result in. But when I do this, I can actually uncomment this right now and run it. It should work. Yeah. This is the Excel file. So it actually looks a little bit different. See, the CSV gives me all this additional information, whereas this is just the plate itself. So you have to make you do have to make a decision which format you're going with, and then write your code accordingly. I decided to go with the CSV. But just as far as loading data into R, this would be an example of how you would do it from an Excel file. All right, so like I mentioned before, we do have to supply the plate layout. So this is how we create that data frame. And if we look at that data frame, like I said, it has rows A, B, and C, and then genotypes, tau ag, EGFP1, EGFP2. So we start by creating new columns that parse this well position into a row and a column. We then only select the relevant data, luminescence, row, and column, and we join the two data frames together. Let's see how there's a column called row in the luminescence data frame, and there is a column called row in the plate layout data frame. I'll open them up next to each other. Actually, I have to do it in here because I didn't say it. Okay, so there's a column called row in the luminescence, and there is a column called row in the layout. And so it will use row as a way to match this genotype to all the rows that say A here, and so forth. So this gives us this resulting data frame where all the rows that say A now say tau ag as the genotype. All the rows that say B now say EGFP1, and all the rows that say C now say EGFP2 as the genotype. Next, we're going to group this new data frame. That won't change anything, but now you'll see you have two groups that are based on genotype. So just behind the scenes, the data is now grouped. And so now when I do the means, like I mentioned earlier, it'll happen within groups. So we've added a new column called mean. Do the same with standard deviation. We've added a new column. Now we take the standard deviation, divide it by the square root of our n. We get the standard error of the mean. Now this is optional, but you can do it. We can now, by assigning this whole pipeline to a new variable, we can now save our tidy data in a new variable. So if I do this, I call it tidy luminescence, and I look at tidy luminescence, this is what it looks like. Right. It's the same as, obviously the same as what we just done, except now I've assigned it to a new data frame, and you'll see it over here, luminescence underscore, sorry, tidy luminescence here. Now we have this. This is just if you choose to do that, and if you want to save this tidy data for some reason, maybe you're doing multiple things with it and you don't want to do the processing pipeline every time, save it as a new variable. For now, though, I'm just going to pipe it into ggplot. like so. We have our discrete x-axis. We have our y-axis. We add the bar graph where, because we supplied the individual data points, we're going to summarize them and only display their mean as bars. We're going to add the error bars that will extend from the mean minus the error to the mean plus the error. It looks like this. We changed the axis labels, graph title, and overall appearance. You see the data hasn't changed, it just looks nicer. And then now we're going to do the one-way ANOVA with the stat compare means, method equals ANOVA. And I want the text to be all the way to the top here, so I'm going to specify the label to go to the Y position of 3700. So it goes up. I do this because I know there's going to be comparison brackets here, and I want to give them space. And I'm going to do my pairwise comparisons in the end, and I specify a list of comparisons that is the control group to GFP1 and the control group to GFP2. And that is what that looks like. So before I execute a command that does it all in a normalized way, I want to make one thing really clear. During this entire course, we've done everything very iteratively, right? So you see, we've started with just one line of code, and then we added another line, and we added another line, and we added another line, and we rewrote everything before every time. The only reason we did it this way is to show you guys how the code works, to show you step by step how the analysis works. It's really important to understand that you don't need to do this when you actually write your code. In fact, you shouldn't, because you're writing these first 3 lines like 16 times unnecessarily. All you need is this block of code. Does everything, right? You don't need to do this iteratively. The whole point is you can do one giant pipeline. One more time, the only reason I did it line by line is to show how you build this pipeline. But you can just execute this block of code and it'll work. I'll show you that. I'll prove that to you in a second. So here is the entire pipeline that I showed you in the last slide of the PowerPoint, where we have this mutate line right here. That's the crucial one, where we normalize all of our luminescence values to the mean of the genotype group, and then just adjust the mean SD and SEM calculations, and we get this normalized graph. So if you open your luminescence, where is it? _short.r. Open this script. I will prove to you that you do not need to do this iteratively. So here, all I have is 2 code blocks. I read the data in first, and I have one thing that makes the plate layout. Then after that, all I have is 2 code blocks that goes from the very start to the very end, once for the raw data, and once for the normalized data. At the end of each, I have a ggsafe. It saves this as raw_luminescence and normalized_luminescence. You will note this ggsafe does not exist in the long form that I just showed you. So if there is anything called raw luminescence and normalized luminescence in my folder afterwards, it came from this script that did not do it iteratively, that just did 2 large blocks of code. So read in the data, specify the layout, 1 graph, 2 graphs. This is the exact same code that I just showed you, except not line by line. I go to my model 6 folder, I open raw luminescence, and I open normalized luminescence. This should prove, beyond a reasonable doubt, that all you need to start from the raw CSV file to get to this graph right here is 30 lines of code. That is the power of R. All right. That will wrap up all the material that I have to teach today. I'm happy to take any questions about this or anything related or unrelated, really. Any comments that you have about the class, my teaching in particular, the class in general, we'd be happy to take your feedback so we can do this better in the next iteration. And just like to thank you all for sticking around for so long and hearing me out.
Thank you, Simon. This is great. I also just wanted to really quickly say kind of what the next steps you can pursue are. So we offer, we've offered in the past a workshop on RNA-seq analysis. This is kind of like the sort of beginnings of the skill set you would need to have in order to do RNA-seq analysis. So if you go to our website, you'll be able to see that there. I also, actually, Simon, do you mind if I share my screen really quick? Here, let me go ahead. So are you guys seeing my screen? You are? Okay. I feel like I never get this to work right. So 1 second. Okay. So if you go to our website, this bigbioinformatics.org, you can see that R and RNA-seq analysis here. So basically you would be starting from week 6. Okay, so this is where essentially you've already gotten the tidyverse skills that you need, and you start learning how to process some RNA-seq data using R. So this is an excellent place to start. Now, the other thing which you can do is learn single-cell RNA sequencing. We have a workshop that was actually just, we ended it just a month and a half ago or so, where basically you can start at the point you're at now, and you can basically move on to analyzing single-cell RNA sequencing data. We're also going to have one in the fall that's going to be all about how you can use the skills you just learned in order to analyze epigenomic data. So that's ATAC-seq, ChIP-seq. We're probably going to even get into a little bit of GROW-seq towards the end. So it's going to be a really fantastic workshop, and you're basically all qualified to participate in it if you've made it to this point. The last thing that I'll plug is unrelated to big bioinformatics, but if you're looking for ways to grow these skills and actually apply them working on real world academic projects, there's something that we just recently created, or I recently created, again, unrelated to big bioinformatics, called Bioinformatics Research Network. Basically, this is where you can come and you can find some projects that have been offered for open source collaboration. If you've got the skills and if you demonstrate the skills necessary to join one of them, you're able to join it, participate fully as an equal member. These are serious academic projects that are intended for eventual publication. So if you are a contributing member to one, once it gets published, you would be an author on it. And so that's a really great way not only to grow your skills and get real project experience, but actually to have something to put on your CV if you're trying to break into the bioinformatics field that says, I actually have worked on one of these projects before, I've been a collaborator before. So if you're interested in this, feel free to check out the website and subscribe to stay tuned for all of our future workshops, and especially for the upcoming epigenetics one. So I just wanted to plug those things before we took questions and feedback.
Great. The whole thing was really nice. Thank you so much. Appreciate it.
Thank you.
Yeah, I think I learned quite a bit from this, although I'm a little behind on my data camp, but I do intend to finish all of my project. I just wanted to ask, so let's say somebody who's done this workshop of R, and how long do you think it takes to, practice and refine and get better at it, and then the next step towards, like, I'm interested in learning Python as well, but I just want to get to know from you guys, is that making that jump from R to Python, what it takes, what needs to be done in between? Just practice more R?
So, do you want to take this, or should I?
Well, I guess I'll say just so we can release anyone who's not interested in feedback or questions. You're free to go if you don't want to stick around. Just so everyone knows. But yeah, no, I actually started with Python and then switched to R. So it's kind of hard for me to to say what it would have been like the other way around. I don't know. Simon, did you also start with Python?
No, I started with R. They were pretty close together, but I did start with R. Well, I started with C all the way back in undergrad, but I've forgotten just about all of that because I never used it. So it's kind of like asking, should I learn French or Spanish first? They are related languages, and knowing one will help you learn the other. But it depends. Do you want to do an R project first, or do you want to do a Python project first, right? They're obviously not equivalent in every sense, but they can do a lot of the same things. You can do this, what I just showed you today. You can easily do that in Python.
Sure.
And vice versa. Okay. So if your goal is to mainly use Python in the future, then I would say get started on Python today.
Okay.
If you want to be a hybrid person, then learn it when it props up or whatever. I will tell you that, well, Henry will have a lot more experience on this than I do, but my sense is that a lot of academia is a bit more R-centric and a lot of industry is a bit more Python-centric. This is not a hard and fast rule. There's plenty of industrial applications that use R, and there's a good number of academics that use Python, but just, the means shift that way.
Sure. So my ultimate goal, I want to be able to use these things. I want to be able to use these use these tools to do skeleton-based image analysis. And I know that's like a goal that's way too ambitious at this point in time, but that's where I want to be. And I just want from you guys, what's the path that I need to take in order to get there?
This is so funny you should mention that. Something I really should have plugged but didn't, because I was talking to people I assume were only interested in R. We've got a new workshop starting in about a month and a week, where we're going to actually be going through with a new faculty member at UTESCA who actually does basically pure machine learning. We're going to be going through introduction to Python. We're going to be going through Python for data science, basically, and then we're going to be going through step-by-step, how do you do machine learning workflows, culminating in biomedical image analysis. So I think you might actually be quite interested in that one.
I'm very interested in that, because that's really the ultimate goal that I want to establish.
Yeah. So, I mean,
we'll be advertising probably starting today or tomorrow. Yeah,
you should definitely sign up for that. It'll be very similar to this. It'll be DataCamp based. Henry and I are doing the intro to Python and the Python for data science, so you'll recognize a lot of the sort of concepts, because Python has a data frame just like R does. It doesn't come with it natively. You have to import it, but whatever. It doesn't have something as cool as tidyverse, but you can do really similar things. So a lot of the concepts will be very, very familiar to you.
Sure.
And then we'll go into machine learning. I would say more generally, if there hadn't been such a nice workshop planned by accident right now, if you have a specific application in mind, look up people who do this and look up what tools they use, right? So like, say you want to do biomedical image analysis, go find a lab that does this, read their papers and see, do they use R, do they use Python? What kind of packages do they use? And then go look for online tutorials on those techniques and just, check out what the masters use and learn those tools.
What I've seen for the most part is MATLAB.
Yeah, that's another one.
Yeah, that's another one which I didn't want to even talk about right now. It's like, okay, I don't want to jump far too ahead of myself. So those are two big shoes to fill in. But yeah, I'm very interested in this workshop. For sure, I'll sign up. Thank you.
Yeah, you should do that. I personally was forced to deal with MATLAB for one semester in college, and I have vowed never to touch it again.
Same.
So I'm not the guy to learn MATLAB from, but there is a very avid MATLAB community that swears by it. It is very powerful for some things if you're so inclined. To me, it's witchcraft. But
I agree. And I think the best rule of thumb is to use the tool that is designed for what you're trying to accomplish, right? So if you're doing some sort of like electrical engineering stuff, probably MATLAB is genuinely the best tool. If you're doing some sort of like software development or like back-end web or like just general sort of, basic machine learning, both R and Python are good, but Python's probably best for most of those things.
Okay.
pure stats and data analysis, R is probably the most efficient you can get. Certainly bioinformatics is the most efficient.
If you want to do bioinformatics, you want to do sequencing data, stuff like that, R is the way to go because they're dedicated packages that are extremely well designed, extremely robustly coded, extremely well documented and updated, that are meant just for that. Just because academia is a little bit more R focused, the gurus of sequencing have designed all their stuff and published all their stuff in R and made it available.
Yeah.
So you guys are pretty familiar with ImageJ, right?
ImageJ?
Fiji, ImageJ, like have you any experience? I just wanted to know the macros that are inherently built in within ImageJ, what language do they use? Are they built up on Python? Is that accurate, or am I completely off?
So I helped someone once try to set up a macro in ImageJ. You can actually do multiple languages. So I think fundamentally, like under everything, I think they're based on Java, but like you can you can program them with, I think, Python and JavaScript, if I recall correctly. I don't know why you do it with JavaScript, but Python, I think you can definitely do it with Python.
Okay. So the answer that I'm getting is I need to learn Python. So I think I've got my answer for that for sure.
If you're serious about if you want to become a professional bioinformatician or data scientist or any of these things, any sort of computational, you really need to have a language that can do the thing that you'll be doing most of the time. So for me, that's R, bioinformatics. But you also need a language that is less specialized, that you can use for more general purpose things. Like for me, that's Python. I need to know both to be effective. If you get into that sort of professional sort of role, you'll probably find something similar. Having both is very good.
And you will learn if you focus on R, like if you want to be who Henry is, right? You focus on R and deal with Python when you have to, you'll get good enough at Python to do the more general stuff. But like put your focus on where you're doing the complicated bit of your work, right? Because like I said, it's kind of like Spanish and French. If you're really, really good at one, the other one is not that hard to pick up. So a bit of Googling, you'll get there, right? So yeah, take the Python workshop this summer. You still have access to the academic data camp through this workshop for, I don't know, a few more months. And that includes all of data camp, which has an enormous amount of Python content that is really, really well done. I actually learned Python through data camp myself. I went on the Python for data scientists like career track and just started from the top and kept going. I'm like, I don't know, halfway through.
Okay.
It'll keep you busy. It'll teach you a lot.
Yeah.
Okay. Thank you.
So were there any other suggestions, any feedback? Adam asked, I'm so sorry, I completely missed this. Adam asked, will the videos be up forever? They'll be up forever. We have no intention of ever taking any of our material down. And as far as I know, YouTube will be here till the end of time. So we'll see however long YouTube exists.
Yeah, YouTube will probably be the reason for the end of time.
Perhaps, yeah. So yeah, and I mean, I think all of us are very committed to the idea of making these training materials and teaching materials as freely available as they possibly can be. In fact, I think we're probably going to switch all of our videos at some point to be Creative Commons licensed so that there's like truly no barrier at all. But yeah, we'll talk about that again at some point.
Yeah, I mean, as someone who's recently used these workshops as a primer and then data camp to learn how to code, such as my level is now, it is so valuable to have this stuff really available. I mean, data camp's great, but it costs like $120 a year, which like isn't that much, but it's $120 a year. Not everyone has $120 a year. And I would have been very hesitant to spend $120 on something that I wasn't sure I was actually interested in yet, right? I just wanted to try it.
Yep.
So having access to these resources that Big Bioinformatics is providing freely now, and a little bit extra because we get access to data camp through it, it's just, yeah, it's invaluable.
Yeah, I think for me, a lot of the motivation is that there's, especially just knowing, there's a lot of places in the world where the amount of money that data camp costs is absolutely untenable. And so if you want to learn bioinformatics, if you want to learn data science, even spending that small amount of money, $10 or $15 a month, is probably not something you can do. And therefore, like, having resources that are cheap is not as important as having resources that are free. And that's why I think for me, a lot of the emphasis is on making sure it's completely free, completely open. Okay, so are there any other questions, any feedback? We're really, this is always a new process for us. We started this less, I think, than a year ago now. So we're still learning. Anything that you can provide, even if it's critical, it's always appreciated to help us improve for the future. Okay.
Sorry to interrupt. Yeah,
Sure.
just feedback-wise, overall, I've like overwhelmingly positive things to say about this course. I think it's absolutely fantastic. But for the data camp modules, when you got into the second week, which kind of introduced you to sort of the higher concept portions of R, we were given like a week to complete that module, and the step up from the first week was pretty substantial. I'd say I really kind of struggled to find the time after a pace was set to get that done within a week.
Absolutely. Yeah, I apologize about that. We kind of realized because of this and because a previous workshop where we experienced something similar, that in the future, what we really need to do is give like a month or longer for people to complete like a couple of those data camp modules before the workshop actually starts. And that way, when you come to the workshop, you're coming with that information already ready, and you don't have this time crunch to finish a 16-hour course in like a week or two.
Yeah,
great.
Yeah, I appreciate that feedback. That's like Henry said, that's the sense we've gotten. It is that activation energy of getting into R. You need to get over that big initial hump of understanding all the data structures, understanding the control flow, understanding all of that, so that later down the line, when you do the tidyverse and suddenly someone does a factor because it's discrete, you're not like thrown for a loop, but you remember, oh, right, that's how a vector works when it's discrete. So yeah, it does come with that initial activation energy of just having to slog through our basics.
Yeah, well, as I said, in many ways, it was actually what kept me going was kind of like the deadline to get it done by, and that kind of kept me on track. It's just that the second module was, I felt so much bigger and so different compared to everything else that I felt like I needed more time compared to the other ones.
Absolutely.
Yeah, we'll keep that in mind. Thank you.
Yeah, I think I would concur with Adam on that one, because the first one I was able to get through in the time frame that was allotted, hypothetically, but the second one, it just took me almost more than my entire working day to get through 3 modules. So I was like, oh man, this is taking far too long. And then it reflected that I'm late to submit. So it sort of negatively reinforced the idea that, okay, I'm late for my submissions, I'm falling behind. Plus I have to do like, 10 different things in the lab. You guys are aware of that. So I guess having a little bit more time, or maybe not making it far too anxious. This is something that's extremely personal, but since you guys asked for some feedback, I'm just letting you guys know that I was getting a little anxious, like, oh man, I haven't got to the modules. But then when I spoke on the thing that these things are not going to be evaluated, we can do it at our pace, I was like, okay, I can get to them whenever I find the chance to, but I made sure at least I'm following up with you guys on each class so that I'm not falling off too far behind.
To be honest with you, some of the stuff in that intermediate R course, wasn't really necessary. Like the apply family, not necessary.
Yep.
Even the for loops and stuff like that. Like I can count on one hand the number of times in the last month I've written a for loop, because once you get good at R, you don't write them anymore, because it's probably the worst way to write code in R. Yeah. So maybe what makes sense is doing that introductory R part and sort of doing a couple things from the intermediate R, but then more getting directly into the tidyverse, because I think that's where you have more fun anyways. And that's where it's certainly easier. Okay. I don't know. Would you agree with that?
Yeah, I think I could agree with that. I will say that even if we're not going to use the for loops, it wasn't like a waste of time. It was still good to wrap your mind around how code works in general and how R studios. So I wouldn't maybe be so quick to discount it, but the tidyverse stuff, it was so much more fun and definitely more engaging, I feel like.
Okay. Yeah. Well, it's the for loops and the if else. If you do, well, yeah. I have like a whole thing about vectorization in R, but basically, if you write vectorized code, you actually won't be writing for loops. And if you use the tidyverse constructs for everything, you actually would never use if else. But those are such fundamental programming concepts that you would get to in the first few weeks of any self-respecting programming course that it feels wrong to leave them out, And
it's also, if you're going to go out and Google stuff in the wild, you're going to run across them like
Oh,
people
yeah.
are. So I feel like it's kind of doing people a disservice to pretend like they don't exist.
That's true.
De-emphasizing them is good. But I sort of concur with Adam that we shouldn't be too quick to entirely dismiss them.
Yeah, that's true. Maybe while loops we can dismiss a little bit more. I legitimately only use while loops like once or twice ever.
Hey, remember that tic-tac-toe? I made liberal use of while loops.
That's right. Yeah, we had this thing where we asked people to make a game of tic-tac-toe using R, and that is one of the few places I've ever used a while loop in my life.
And not even a very complicated one, just while true.
Yeah.
Just speaking personally, I think like if for my ideal kind of learning pace, you know how we had that review week for the third session?
Yeah.
I maybe would have said like in data camp, do conditions, loops, and functions for the first 2 weeks. And then for the 2 weeks following that, before the review week, do the apply family and the utilities. So same stuff, but just broken up and over a large period of time. And that would have been perfect.
Mm-hmm.
Absolutely. Yeah, no, that's a really good point. And it's so easy to break that up too. Yeah, we should do that next time. Yeah.
Other than that, though, this is absolutely fantastic. Really appreciate the resources you've given us and all the lessons you've given us as well. Thank you.
Yeah, thank you.
You're very welcome.
Hey, I have a sort of a school related or like a professional career development related question. So if I'm to add this workshop that I've attended, all the modules that you guys delivered, I wanted to add this to my CV and resume. What's the best way that you guys recommend that I add this?
That is an excellent question that we have discussed behind the scenes.
Yeah. So
go ahead.
Okay, so it depends on for what purpose, right? So what is it you would like to demonstrate?
One that I have at least taken this workshop, I'm familiar with these concepts, and then based on that, I want to be able to finally have at least like one statement saying that I've used these techniques or used these tools to apply and use it for my dissertation research. That's what the overall goal would be.
That's what I did right here.
Okay. Okay.
Yeah.
It's not polished because I'm not, my sort of approach to CV is yeet it all on there and then delete stuff to tailor it to whatever you're applying and then refine. But
okay.
But you're talking about something that can demonstrate that you have that knowledge. I mean, I personally, from my point of view, that's part of why I made this bioinformatics research network thing, because a lot of people, you learn something, but you have to apply it before you can demonstrate that like you actually know it. You have to do a project and you have to get published, or at least you have to be working on something you've got on your GitHub,
right?
Something like that. So that's part of why I made this. I would recommend checking this out. Because I think if we create, like we were talking about, like making a certificate or something, basically saying we certify you've completed this thing. But if I'm a recruiter, that will mean literally nothing to me. Like I've read a lot of CVs, and for people who wanted to do something bioinformatics related, when I read their CV, I literally just skip right to the project part. So I don't know, maybe other people are different, but for me, that's how I read CVs typically. So I think having a project is the important part.
Okay. Okay. Yeah, that makes sense.
Yeah. If I would concur that I'm actually currently doing the trainings. I've just finished up a large portion of the trainings I need to do for the Bioinformatics Research Network. So if you go on there, here, can I share my screen real quick and show the project page? If you look at this right here, whoops, come on. So this is the project page, right? So there's a few projects, and then I go to the RSeq one here.
Okay.
I'm going to jump on that probably. It says a little description, blah, blah, blah. And at the bottom here is training requirements, right? So you go to R programming is one of the things that you need to do. Okay, let's have a look. Sends you to the GitHub. And here it says code a game of tic-tac-toe, which is the one time you're going to use while loops. And then, there's a bunch of instructions and so on and so forth. But once you do those trainings, you get to be on this actual academic project, right? That's it's currently in late stage. It's targeted to be published around December 21. Just to give you sort of an idea of the time frame that you can be expecting from this. So I'm probably going to hop on this because I want something on my CV that is honest to God work that I can point to that's going to be sitting on my GitHub.
Yeah, well, just to clarify, it's a bar you have to clear before you could join that project, but the team still has to meet and decide, if they want to bring you on, because the team has control sort of over who gets to join and who doesn't. Because it's supposed to be like egalitarian, democratic. I would never force someone to join a project or force a team to accept someone. Right,
but. Yeah, so that's kind of my path because I'm facing a similar issue as you. I'm looking to make money off of this. So I need to show that I actually didn't just sit in on some Zooms, but actually did stuff, right? Currently, if you were to put a gun to my head and force me to apply for a job right now, I would do what I did on my CV. I would have that, and then projects. I would say something about RNA-seq that I did during that workshop, and I would just make it look good. And then in my cover letter, I'd say, I have developed a strong interest in this. I have striven to go beyond my curriculum and took this workshop, and in the course of it, we have done a bioinformatics analysis, and I'd be happy to show you.
Okay. Because I was going to say
ideally, do the thing that gets published.
Say that again.
Ideally, do the thing that gets published because.
Yeah, absolutely. So what I was gonna get at is the certificates. I'm not too worried about that because each time I do the data camp modules, I get a certificate. So that's a good thing because I can save those. But the other thing was just like you guys mentioned about the workshop with Python and machine learning. That's where I have a boatload of images that I do wanna push through, a Python script. That is able to generate data, and that I am going to be able to use for my dissertation, my publication, and it's a thing that's going to stay with the lab. So anybody who comes by it has to, I have images, here's the script for it, plug it in and be done with it. Nobody has to do anything apart from that.
Yeah,
that's where my goal is. I think it's going to take me maybe about 6 to 9 months to get there, but that's what I'm aiming at.
Make sure from the beginning, make sure you put it on GitHub. I check when I'm checking CVs, I always check people's GitHubs. Because that's kind of like an unofficial CV of what have you actually done related to some sort of informatics or data science.
Okay. And let's say if I want to collaborate with if time and scheduling permits and if interest permits as well, let's say I want to collaborate with the big informatics group to get help on for my own particular project, what would be the best approach you guys would suggest? Best approaches, best people, I guess.
So I'm just going to show this really quick. So basically with our research network, so this is again not part of Big. Big is totally separate from this. Right. This is just something I'm also involved with, and also Simon's also involved with, but different organizations.
Sure.
So basically you can sign up. We've got a sign up form here. Basically, if you complete this out, you'll get a link to join our Slack channel. So the Slack channel, you'll get all the information. You'll get a chance to, talk to people who've got projects. But basically, that's really more to provide a community. The way you actually would begin working on a project is you would go to the projects, you would find, I don't know why this is not working. You find one or two that interest you.
Okay.
Complete the training. So basically, if I see, this one requires some, I don't know why this is being slow right now. Okay. Well, whatever. So basically, okay. Yes, thank you, Wix. You figured it out. Oh my gosh. So what I get for trusting a pre-made website maker thing. Should have just made it myself. Okay, well, anyways, if you go projects, okay, whatever. So each of them will have the training requirements, and basically, yeah, so here it is. So basically, you'll complete whatever these are. So if I want to do this one, I would come to the GitHub, I would complete the instructions here. Okay. And if you're part of the Slack group, basically, when someone finishes a training, they just let me know and I'll check it. And if everything's complete, I'll say, okay, complete. You finished that training. And then once they complete all the ones necessary to join a project, then I'll basically advocate for them. I'll go to that project team and I'll say, hey, this person's completed all their trainings. They're interested in joining the project. Can they come to a meeting? And they'll decide, whether or not to let you come to a meeting. And if you come to a meeting, they'll, talk with you about the project. They'll let you know about it. They're very nice people. They're not going to really be exclusive, I've found. And then you guys can decide together whether it's a good fit and whether you want to join.
Okay, cool.
But yeah,
I'm about to go through that process, so I'll let you know how it turns out for me.
Yeah, sounds good. I'll come talk to you.
All right, cool.
Thank you so much, guys. This is really helpful.
Yeah, you're welcome, man. Happy we could be of help. Happy we could actually be useful with this to people. Because I would have done it either way, because it's made me a better programmer, just because I have to learn things to teach.
That's good. It's awesome.
Good to know that people have gotten value from it.
Exactly. Okay, so I'm sorry, I don't know is your first name E or I? So you said we can still reach out to ask. Yeah, you can always reach out. We're here to help. And I'll at least be around for a few more years. Simon, not so much. Simon's going to move on to bigger and better things, but I'll be around. You can always reach out. All right. Are there any other questions? Any other comments or criticisms before we break? Okay. Well, I just wanted to express I was really I thought this was a fantastic idea for a workshop, and I'm so glad that you have all stuck to the end of it. I'm really looking forward to seeing you all in our future workshops, future events, and make sure to subscribe to stay up to date with future goings on. And if you're interested in coming to that Python workshop, especially subscribe, because we're going to send out an email probably today or tomorrow to everyone who is about how to get enrolled in that. And with that, I don't know, Simon, anything else you wanted to say?
I'll just thank you everyone for sticking around, for asking questions, and for being here. It's been lovely.
I totally concur. All right. Well, with that, thank you all so much, and hopefully I will see you again in a future workshop. All right. Goodbye.